this video we will discuss about the T cells from iPSC has more anti-tumor response than gamma delta T cells. So in this video we will discuss the preliminary uh, results that shows the T cells that are differentiated from iPSCs. And the results for its comparisons with gamma delta T cells, we will discuss that thing in our next video. In this video we will discuss what are the experiments that they are planning to compare this hypothesis that has been published in previous literatures. So for that, first they developed a CAR construct that can target the ovarian cancer antigen, that is GPC3, and for that they use the GC33 single chain fragment variable antibodies. And the idea is that they took the CAR cells and transduced them, and they took the CAR cells and they took these iPSCs and transduced them with this CAR construct that is mentioned in the panel E, and then do the homeopathic inductions towards the T cell lineage. And they took the two different populations, first the immature CAR-T cells, and directly expand them using the antigen to activate these T cells, CAR-T cells, and that will cause the expansion of the CAR-T cells. And in other case, what they did is they allow these immature CAR-T cells for the maturations, and thereafter they use those CAR-T cells for the functional studies. And in the third case, they took these gamma delta T cells from the PBMCs and then expand them and then use those PBMCs CAR-T cells for the functional studies. And the basic markers that they used to differentiate these cells, that is mature CAR-T cells, that is they have the CD5 and CD8 beta positive signals. So they sought the CD5 and CD8 beta positive signals in order to get the post-maturation iCAR-T post-maturation cells. And the pre-maturation cells, that is the cells that they took it just after the homeopathic differentiation, homeopathic progenitor cells differentiations or inductions into T cell lineage. For the PBMCs, the sorting criteria that they used is CD8 beta positive cells, and use these three populations for the functional studies to compare which of them has more anti-tumor response. And thereafter they also in order to check what are the different markers, or in order to further validate the immature iCAR-T cells and the mature or post-mature CAR-T cells, they check it using the different markers, that is CD3 and CD8 markers, and they are able to see that some of the populations in mature CAR-T cells has both the markers, and they also check it using CD7, CD8, CD5, CD8, CD4, CD8, and CD8 alpha and CD8 beta. And similar results they did it for post-maturation CAR-T cells. Although in most of the cases the results are similar, in some cases we can see the results are different. For example, the case where it shows this CD62L and CD8 beta, these cells are immature cells are more positive in when both the populations are considered, whereas for the mature cells they are negative. And in other case they are positive around CCR7 and CD8 beta. That can be seen for the post-maturation CAR-T cells, but they are negative in the immature CAR-T cells. So they use these different markers validations to further validate the immature CAR-T cells and the post-mature CAR-T cells. So this is how they plan to compare the fact that is, or compare the results that is presented in the previous literature, that is T cells from the iPSCs has more anti-tumor response than gamma delta T cells. They will compare these three conditions and then check whether they are able to see the results that is in line with the literature, or they see the different results. We will discuss the results of these three conditions in the next subsequent videos. I hope you understood how they uh, develop the experiments to check their findings that has been previously published. So if you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.